Hello, I'm Alex Blumendahl. This is my first Boulder workshop. And like Ben, I'm disappointed not to be joining you in person, but I'm thrilled to be here with you virtually now. So let's talk about genetic variation in the population, how it arises and is maintained, how it's distributed and what forces shape its distribution, how it's characterized and quantified, and ultimately how these aspects impact our efforts to understand its effects. I'll share a list of references alongside this lecture, which I encourage you to check out. We might begin by asking how much variation there is among our genomes anyway, or indeed among the genomes of any species, in some absolute sense. A natural way to quantify this is to ask about the average number of differences between two people's genomes. A precise notion is that of nucleotide diversity, due to Ney and Lee. If you sample two random gametes or haploid genomes from a population, what fraction of nucleotides differ between them in expectation? Nucleotide diversity is typically denoted pi. Multiplying by the length of a genomic sequence gives the expected number of differences. To see how it might be estimated, let's think about a single site where there's a biallelic single nucleotide variant. If the frequency of one of the alleles is p, then the probability that two random copies in the population differ is 2p1 minus p, a quantity known as the expected heterozygosity. Given an estimate p hat from a sample containing n copies, Ney showed that an unbiased estimator for expected heterozygosity is this quantity h hat, which is n by n minus 1 to 2p hat 1 minus p hat. This quantity is the probability that two distinct copies differ in the sample. Anyway, you can then just average this quantity over many sites to obtain an estimate pi hat. Nucleotide diversity in humans is estimated at around 1 in 1,000 or 0.1%, or a few million sites out of our 3 billion. This figure has held steady for about 30 years, despite our ability to sequence ever more genomes ever more deeply. The value varies a bit depending on the ancestry of the individuals sampled, but not much, as we'll see later. Our closest relative species, the great apes, chimpanzees, bonobos, gorillas, and orangutans, have very similar pi values, with orangutans the largest, but only about twice that of humans. As for some of our commensal species, mice and rats' pi values are around 0.2% as well, with fruit flies coming in a bit higher at 0.8%. For some more extreme examples, the cheetah's is only 0.02%, while the hyperdiverse split-gill mushrooms is as high as 20%, apparently the record holder for eukaryotes. Now, you may have heard that given mutation rates and the size of the human population, every variant compatible with human life exists somewhere in some individual. And while that may be true, the vast majority of these variants are exceedingly rare. Heterozygosity emphasizes common differences or polymorphisms. And a classic, if arbitrary, definition of a polymorphism is that the less common or minor allele has a frequency of at least 1%. So why do we have genetic variation in the first place? The ultimate origin is, of course, mutations. But how does a variant reach an appreciable frequency in the population? What forces influence allele frequencies and their distribution? Now, the first thing you might think of is natural selection. As Darwin taught us, mutation and selection together are responsible for life as we know it. But scientists at the turn of the 20th century realized that it was also necessary to explain how variation is maintained in the population. Their fundamental ideas were inspired by a rediscovery of Mendel's laws, and they make sense even in the absence of selection. This so-called neutral theory provides extremely important intuition, and in fact models a majority of our common variation to a good approximation. It therefore serves as a kind of null model. It further serves as a basis for inference. To the extent that the variation we carry is shaped by our ancestors and their demographic history, the theory allows us to make inferences about our past from observations of present and sometimes also ancient genetic variation. We'll therefore spend some time on the classical Wright-Fisher model, dating back to seminal works by these two authors around 1930. In the simplest version, a population of haploid alleles segregating or polymorphic at a given locus evolves from generation to generation. Time is discrete, generations are non-overlapping, and for each new allele in the next generation, a parent is chosen independently at random from the current generation. This is an idealized model of random mating, and also of Mendelian segregation if the haploids are paired in diploid individuals. I've drawn an example schematically here. As you can see, some alleles are copied once, some more than once, others not at all. There are 3 copies of the alternate allele in the current generation and 4 in the next. These dynamics describe a Markov chain, a stochastic process that can be continued indefinitely, and the number of copies of each allele will fluctuate from generation to generation. We'll assume no further mutation at this site. That's called the infinite sites assumption. Now, if a certain allele has frequency p in the current generation, how is its frequency distributed in the next generation? Well, each copy of the allele in the next generation is a coin toss, an independent Bernoulli p random variable. And if the next generation has size 2n, we usually write 2n because we're diploid, the allele count is binomial 2np. And that means that the new frequency p prime therefore has conditional mean p and conditional variance p1 minus p over 2n. In this way, the frequency will evolve as a Markov chain in its own right, a sort of random walk driven by the underlying binomial sampling. This phenomenon is called genetic drift. Note that the rate of drift is proportional to heterozygosity and inversely proportional to population size. I've drawn a sample path here, showing how the allele frequency might evolve over time. And in this neutral setting, the conditional expectation of the allele frequency, it never changes, even if the population size does. In probabilistic language, the Wright-Fisher process is a martingale. This is an important property. The allele frequency might fall to 0 when we say the allele is lost, as in this example. It may instead rise to 1 when we say the allele is fixed. These are absorbing states. They're permanent because we assumed no recurrent or back mutations. Now, if the population size is fixed, say at 2n, the allele will eventually be either fixed or lost. And conditional on the current frequency p, what is the probability that the allele will eventually be fixed? Well, by the martingale property, it has to be p. So a brand new mutation starting at p is 1 over 2n will have only a 1 over 2n chance of eventual fixation. So the odds against are 2n minus 1. Those are pretty long odds in a large population. But time and the genome are long too. If you throw enough mud at the wall, some of it'll stick. Sometimes it's not survival of the fittest, but merely survival of the luckiest. Neutral theory has become foundational to how we understand evolution. So genetic drift acts to remove heterozygosity over time. Another way to think about this is that heterozygosity, h, is a concave function of allele frequency p. So the expected heterozygosity after some time or after some drift is always less than heterozygosity of the expected allele frequency, which stays put. To make this intuition more quantitative, a bit of simple algebra shows that with h prime is 2p prime 1 minus p prime, the heterozygosity after one generation, you have the conditional expectation of h prime given h as 1 minus 1 over 2n h. Now, over many generations, we therefore have this exponential decay. So you can put that slightly differently. The expected change in heterozygosity, expectation of delta h given h, is simply minus 1 over 2n h, which says that the expected heterozygosity is lost at a constant relative rate of 1 over 2n. And if you pass to a continuous time differential equation, you again obtain the same exponential decay behavior. Now, the future allele frequency pt, given the current allele frequency p naught, accumulates variance. And that happens in a complementary way. You can also show in some simple algebra that the variance of pt given p naught is approximately p naught 1 minus p naught 1 minus e to the minus t over 2n. And if you think about that equation for short times, you can use a linear approximation, which clearly generalizes the single generation evolution rule. But in the long time limit, pt tends to the absorbing state, which is actually a Bernoulli random variable. It's 0, 1. It's lost or fixed. And you see the variance of that Bernoulli p naught 1 minus p naught in the long time limit. Anyway, you can modify the Wright-Fisher model in many ways. You could incorporate sexual reproduction for diploids, variable population size, variable reproductive success, non-random mating. But under neutrality, the basic features of genetic drift due to binomial resampling are highly robust. In fact, the only modification generally needed is to scale the rate or variance of drift by a constant. Now, in realistic populations, this variance tends to be somewhat larger than in an idealized population of the same size. That constant is traditionally absorbed into n, and the effective population size, NE, is defined as the size of the ideal population with an equivalent rate of genetic drift. The same evolution rules hold, but with n replaced by NE. So the conditional variance of p prime given p is p1 minus p over 2 NE, and the expected change in heterozygosity is minus 1 over 2 NE times the current heterozygosity. Now, in real populations, that means NE is generally somewhat smaller than the actual population size. Notice that the Wright-Fisher model is so canonical that we actually define key parameters in terms of it. So, if mutation creates genetic variation and drift removes it, how do these forces balance over time? In 1969, Kimura considered this question in the context of the infinite sites model with a constant size population and derived the equilibrium heterozygosity. Here's a brief heuristic derivation. Let mu be the rate of mutation per site per generation. Then new mutations enter the population at rate 2 NE mu. New mutations thus lead to an influx of heterozygosity at a constant rate 2 times 1 over 2 NE times 1 minus 1 over 2 NE, that's the heterozygosity of the newly arising mutation, times 2 NE mu, which is about 2 mu. Now, if you look over many sites, you can get the evolution of nucleotide diversity. The expected change in pi in a generation is 2 mu minus pi over 2 NE. And if you then set that to 0, you get this equilibrium value, which is 4 NE mu. This population scaled mutation rate, 4 NE mu, it's a fundamental parameter of population genetics, and it's often denoted theta. So the result is the expectation of nucleotide diversity, pi, is equal to this parameter theta. Now, the infinite sites assumption is generally a reasonable one. That quantity theta is much less than 1. So in eukaryotes, mu, the per site mutation rate, is on the order 10 to the minus 8. The mean heterozygosity of 10 to the minus 3 in humans, then that squares the long-term effective population size of about 20,000. That number 20,000 reflects our long-term history as a species, as opposed to our recent exponential growth, as effective population size tends to be dominated by the smaller sizes over time. It's a harmonic mean. So that's how that mutation-drift equilibrium works out. Now, in the 1980s, Kingman, Hudson, Tajima, and others developed a powerful new probabilistic approach to analyzing the Wright-Fisher model, known as the Coalescent. Instead of looking forwards in time, one looks at lineages of haploids going backward in time. Now, two lineages coalesce at their most recent common ancestor, and they coincide from then on back. And I'm just going to show you how it works for a sample of 2. Coalescence occurs between any pair of lineages with probability 1 over 2ne in each generation. For a given pair of lineages, coalescence time is therefore a geometric, or approximately an exponential, random variable with expectation 2ne generations. Now, what's more interesting is that the coalescent is totally independent of any mutations that occur, which can therefore be modeled as sprinkled randomly over the coalescent after the fact. To illustrate the flavor and simplicity of this approach, here's a sketch of the argument of the mutation-drift balance result. 2 haploids differ at a site when a mutation has arisen on one of their lineages before those 2 lineages coalesce. Mutations occur with rate mu per site on each lineage, independent of coalescences. So the expected number of mutations per site on the pair of lineages before coalescence is 2 times 2ne mu, which is 4ne mu, or theta again. The nucleotide diversity can be thought of as the mean fraction of sites that are segregating in a sample of 2 individuals. Now, what about the fraction of sites that are segregating in a sample of size little n, which I'll call Sn? The coalescence argument may be generalized to show that the expectation of Sn is theta times 1 plus a half plus a third all the way up to 1 over n minus 1. That's interesting. That expression grows logarithmically. That's very slowly, and it remains a reasonable approximation when it's much less than 1. If you multiply it by the length of a genomic sequence, you get the expected number of segregating sites or variants in the sample data. That result is originally due to Watterson in 1975 in joint work with Wu. And a historical note here is that although this paper would become highly cited, Wu was not given authorship and was not encouraged to pursue independent research, which was unfortunately common practice for female programmers and research assistants at the time. After many years of scientific work and teaching, she did eventually get a PhD in her 40s and is now faculty at the University of Melbourne. So Watterson and Wu proposed theta hat, an estimator for theta, as Sn over this harmonic sum. And that's actually an unbiased estimator. Actually, notice that we now have 2 estimators for theta from a given sample of sequence data, one based on nucleotide diversity and one based on segregating sites. Both can be applied to any given data set for any segment of the genome. Tajima in 1989 actually developed a statistic to compare them and proposed a significant difference as a test for violations of the neutral equilibrium model, violations like selection or changing population size. Anyway, if you investigate beyond the number of segregating sites in a sample of size n, you might ask about the distribution of their allele frequencies. So what's the expected behavior of their histogram? The number of singleton sites that appear just once, frequency 1 over n. Doubletons that appear twice, frequency 2 over n, etc. That object is called the site frequency spectrum, or the SFS. An equivalent question is, how many generations a new allele is expected to spend at each frequency before it's either fixed or lost? The equilibrium behavior in a fixed size population goes all the way back to Fisher and Wright, who showed the SFS is proportional to 1 over p. More precisely, the expected fraction of sites at allele frequency p is k over n is theta over k, which I've represented by area in this histogram. And that result holds in a sample or in the whole population. So in expectation, we'll find theta singletons, theta over 2 doubletons, only half as many, theta over 3 tripletons, etc. As another example, there's only 1/10 as many alleles around 50% frequency as there are around 5% frequency. Now, this law governs the frequencies of newly arising alleles, also called derived alleles, as opposed to their ancestral counterparts. In practice, we may not know which is which. Actually, our best guess for the derived versus ancestral allele comes from, at least for common variants, comes from comparing our genomes with those of chimpanzees or orangutans. It's kind of neat. We may consider simply the minor allele frequency instead. That would make things easier. The minimum of p and 1 minus p, for which you get the so-called folded SFS. Then you just add theta over p with theta over 1 minus p, and you get this law theta over p times 1 minus p. A curious consequence of this form is that the total amount of heterozygosity is actually uniformly distributed over the folded SFS. Now that's because the heterozygosity per variant exactly cancels the relative number of variants. Actually, if you think back to Ben's lecture, what does that imply about the additive genetic variance for a trait? 2p1 minus pa squared in the absence of selection. Well, if there's no relationship between effect size A and allele frequency, then additive genetic variance and indeed heritability are also uniformly distributed over the folded SFS. So to put that simply, you have equal contributions from each minor allele frequency bin. So what factors can influence allele frequencies beyond the basic neutral model? Well, selection. Now, selection comes in various forms, and I'll just briefly touch on a few of them. Perhaps the most obvious is negative or purifying selection. In its most extreme form, a variant incompatible with human life will simply never be represented in the population, and one incompatible with fertility won't last long. In less extreme forms, individuals carrying deleterious alleles have relatively lower fitness, or fewer offspring on average. In fact, selection against a variant can be quantified in terms of the relative reduction in mean number of offspring, which is known as the selection coefficient. Relative is the key term here, both to other individuals and to the pressures of the given environment. On the other hand, occasionally a new allele will confer a fitness advantage. This is called positive selection. There's some pretty interesting examples of environmentally dependent positive selection in humans. Lactase persistence in pastoral farming, adaptation to lower oxygen at higher altitudes, and cholesterol processing in high-fat diets. Note that these selective forces are often talked about as though they're active phenomena, but they're passive forces. They operate purely through relative fitness. A third and more complex example is balancing selection, which acts to maintain more than one allele in the population. The most common form is heterozygote advantage, where the fitness of a heterozygote is higher than that of either homozygote. A classic example of this is sickle cell anemia, a genetic disease that damages red blood cells. The heterozygote carrier, while still suffering a milder version of the condition, is also resistant to malaria infection. So the trait and the corresponding allele are under balancing selection in environments where malaria is prevalent, but under simple negative selection otherwise. The Wright-Fisher model can be generalized to incorporate selection. Alleles now transmit to the next generation with some bias you can write down in terms of the selection coefficient. The martingale property no longer holds. And as for the site frequency spectrum, for a given selection coefficient, there is still a nice analytic solution for the fixed size model, a kind of warped version of that 1 over p law. From this expected behavior, you can deduce various relationships between mutation rate, selection coefficient, effective population size, and allele frequency, relationships that inform our thinking about association studies. Now, I won't have time to get into all that today, but I can recommend, for example, the supplement of Zook et al. 2014 on rare variant association studies. We'll now return to the context of neutral variation to consider the impact of demographic history on genetic variation and allele frequencies. An important scenario is a population bottleneck. So imagine that we suddenly go down to, say, 50 individuals, or 2n equals 100 copies. What happens to the variation? Well, very common alleles at, say, 10 or 50% may be preserved at roughly the same frequency. But 1% alleles, things get a little noisier. It's actually described by the Poisson law. Many of these will be lost. Some may be retained. Some may have their frequency doubled or even tripled. If you go down even lower to alleles at 0.1%, or 1 in 1,000 alleles, 9/10 of these will be lost in expectation. but the 1/10 that survive will have their frequency boosted by an order of magnitude or even more. And in this way, bottlenecks have a profound impact on the site frequency spectrum and ultimately on genetic variation in a population. There have been many bottlenecks in human history, and on the largest scale was the out of Africa migration. That's generally inferred to have occurred 45 to 60,000 years ago, with a fairly stable NE is 10 to 15,000 for a long period before that. Its effects are still apparent in the greater genetic diversity among individuals of sub-Saharan African ancestry compared with those of other ancestries throughout the world. When a bottleneck population founds a new population, this phenomenon is also called the founder effect. And there's various smaller populations around with a pronounced founder effect. Examples include the Finnish, Icelandic, and Basque peoples. Another scenario very relevant to human history is exponential expansion. When the population grows exponentially, the rate of genetic drift declines dramatically, and allele frequencies are almost frozen in place. Because of recent exponential expansion, a majority of common variation in humans is older, or so-called standing variation. In fact, from the point of view of heterozygosity, most variation dates to before the out of Africa bottleneck. As a result, most of our human variation is shared among ancestral groups. So-called private variants represent a minority, as both the loss of older alleles and the rise of recent ones occur much more slowly and rarely in large populations. So speaking of ancestral groups, I'm now going to segue to population structure, which is a key demographic phenomenon I'll discuss for the rest of the lecture. So what happens when a population splits in two at some time? Each subpopulation or group continues to evolve according to the Wright-Fisher process, but genetic drift now happens independently in each group. The result is that the group-specific allele frequencies will begin to diverge, not only from those of the ancestral population, but also for each other. They'll exhibit systematic differences beyond what you'd expect from a random partition of the total population. I've drawn a sample path showing this divergence, and that's of course just one allele, but this is happening simultaneously at sites genome-wide. So population structure, which we might define very generally as any systematic departure from random mating, thus results in systematic patterns in allele frequencies. Now, we haven't talked about linkage disequilibrium. As you may know, genetic variants physically near one another on a chromosome tend to segregate together. And these correlations decay with genomic distance. The reason is that chromosomes break up and recombine at only 1 or 2 points per generation in the formation of gametes and meiosis. Genetic material is thus passed on in long tracts of chromosomes, and the nearer two sites, the more generations it takes for a recombination to break them up and to separate the alleles they carry. Further, recombination events don't occur uniformly at random, but tend to concentrate around so-called recombination hotspots, which leads to a blocky local correlation structure. Now, LD is a core aspect of statistical genetics and of how we think about genetic association studies and follow-up analyses, but the reason I bring it up now is that population structure leads to another very different kind of LD, a correlation structure that's global over the entire genome, even across chromosomes. To get some intuition for this, think about two variants very far apart on the genome whose allele frequencies have both diverged between two groups. If you observe an individual's genotype at one site, you can then make a slightly better guess as to which group they came from, and in turn, a slightly better guess as to their genotype at the other site. Now, the point is that this applies to every pair of sites. Well, to put it another way, individuals in group X carry a relative excess of every allele that is more common in group X. So the simplest form of population structure is discrete, as in the above example. More generally, splits may happen in various groups at various times. The result is that each extant group has its own set of allele frequencies, and these are variously diverged from one another according to the laws of genetic drift. It's natural to quantify the extent of divergence among the groups. And Wright actually proposed an elegant way based on partitioning the diversity in the total population into two components: the diversity within the groups, or the shared diversity, and the diversity among the groups, the diversity due to population structure or differences in allele frequencies. To make this precise, let's consider just a single segregating site in the total population. And we're going to think about expected heterozygosity at that site in a slightly different way. We're going to think about it as the variance of the allelic state of a randomly selected allele, thought of as a Bernoulli random variable. So I'm going to call that variable A. It's a Bernoulli P. And so that variable has expected value A and variance P1 minus P. That differs from the heterozygosity by only a factor of 2. Now, the advantage of the variance point of view is that variance can be partitioned and conditioned, and this is a strong theme in quantitative genetics and linear models more generally. So let's also think of the group identity of the selected haploid as a random variable. I'm going to call that Z. It's a categorical random variable, and these CIs here are the relative group sizes. And so I'm going to write PK for the group-specific allele frequencies. You can write the group conditional allele frequency then as a random variable PZ. And the law of total expectation expresses P as a weighted sum of the group-specific allele frequencies, PK. Not surprising. What's a little more interesting is the law of total variance. You apply that to the variance of the allelic state A, you get two terms: the expected variance of A given Z and the variance of the expectation of A given Z, which works out in this way. Just let you have a look. So on the left, we have the total variance of the allelic state in the population, in the total population. And on the right, we have two terms. The first term is the average allelic variance within the groups. The second is the allelic variance among the groups, or that part due to structure. The fraction of the latter to the total is known as the fixation index, or FST. And Wright used S for subpopulation and T for total. That's where the original notation comes from. The fixation index thus represents the fraction of allelic variance due to population structure. Notice that this fraction reduces to 0 when all the PKs are the same, equal to P, so in the absence of structure. On the other extreme, if the site is fixed within each group one way or the other, though not all the same way since we said it was segregating in the total population, that means all the PKs are either 1 or 0, which means that the first term, the average heterozygosity within, is 0. And in that case, the fixation index is 1. And that's where the name comes from. So the first term, you can also think about it in terms of heterozygosity. The first term represents the mean heterozygosity within the groups, or equivalently, the heterozygosity conditioned on that pair of haploids both being selected from the same group. This quantity is always less than the total heterozygosity, and that ratio is 1 minus FST. This depression of total heterozygosity due to population structure is known as the Valent effect. The balance of total heterozygosity is that part due to population structure. So you'll find various subtly different definitions of FST across the literature, partly because Wright's original definition actually wasn't totally precise. Some authors like to normalize by the heterozygosity in the common ancestral population rather than the total extant population, and that approach has a slight advantage of relating more directly to evolutionary parameters of genetic drift. Well, on the other hand, past allele frequencies cannot be observed directly, and these relationships depend on idealized models of discrete structure that may not reflect reality. Anyway, the approach I'm presenting here presents FST as a simple descriptive parameter of the extant population. Now, of course, drift is random, and each independently or quasi-independently segregating site will have drifted to a different extent. One therefore averages these quantities over many sites. There's actually some tricky estimation issues here, and the literature contains a number of different possible estimators. Without going into details, I recommend Bhatia et al. 2013 for a thorough discussion. How does that actually work in real human populations? So FST values in humans tend to be less than 0.2 at the most for intercontinental groupings, and even values that large appear only between highly diverged groups. They can be as small as 0.001, maybe up to about 0.05 for intracontinental groupings within continents. And in this sense, we can say that the great majority of our total genetic diversity as a species is due to differences within ancestral groups, with only a small minority of it can be attributed to differences among ancestral groups. And that's actually why I said at the beginning that your estimate of nucleotide diversity really won't depend a great deal on whom you include in the sample. So real human populations also feature admixture, where individuals from diverged groups come together as a result of migration or other factors. Their admixed offspring have allele frequencies intermediate to those in the source groups, a weighted average or linear combination with coefficients given by the mixing proportions. These proportions of group-specific genetic material are of course going to be half-half in the initial generation after admixture, multiples of a quarter in the next, a quarter, a half, 3/4, but they'll soon diffuse into a continuous admixture cline. I'm just representing admixture schematically here. And these clines might extend physically over a geographical region into which two or more groups migrated from different origins. So recently admixed individuals also exhibit a special kind of LD, admixture LD, over long tracts of chromosomes. This kind of LD is due to the recombination of previously diverged genetic material. It can persist for 10 generations or longer, and such patterns can be readily observed among individuals of African American and Latin American ancestry. While it's not typically discussed, the definition of fixation index above can actually be generalized to continuous forms of population structure. And without going into detail, there's no need for that latent parameter Z to be categorical. It could parameterize the admixture proportions among the groups by taking values in a simplex, for example. And that's related to the classic model of Pritchard, Stevens, and Donnelly for structured populations. Now, a more complex form of continuous structure is inherently spatial. It's also called isolation by distance. Here, individuals distributed over some geographical region and sometimes migrate to or mate with their neighbors. I've shown this schematically in one dimension here, but it could equally well take place in two dimensions. Drift happens at each location, but now in a spatially correlated way, and the correlation decays with geographical distance. In this setting, allele frequencies will evolve to vary continuously, but not necessarily linearly, with geography. And in the case of spatial structure, the latent parameter Z could parameterize geography. Of course, these are just toy models. Population structure in humans today is tremendously complex, reflecting our rich and complex demographic history. And there's even further kinds of population structure induced by assortative mating on factors other than geography, including even assortment on heritable traits. But a foundational paper in 2006 by Watterson et al. Proposed an essentially model-free way to learn population structure from sampled genetic data. And that's just by doing principal component analysis on a matrix of genotype data. So in detail, you start by selecting some approximately unlinked biallelic sites, generally SNPs. Now, when I say approximately unlinked, I mean that you prune the set of SNPs so that no pair is in strong LD. This turns out to be very important because all PCA does is find directions or components of maximal variance in the data, the best low-dimensional summary of its variance-covariance structure. A block of strongly correlated SNPs will derive a component reflecting variation local to that part of the genome. You learned about inversions of chromosome segments in Ben's lecture, for example, and these can create big LD blocks, as can regions under strong selection. But what we're after here are the much more subtle global patterns of correlation due to population structure. As we talked about before, that kind of LD is spread over the entire genome. So you take these LD pruned SNPs and you form a matrix of standardized genotypes, as I've written down here. You perform PCA on this matrix, and that means doing the singular value decomposition, or equivalently, you can form the sample covariance matrix, which is also called the genetic relatedness matrix in this context, and do an eigendecomposition. What really matters is that you're projecting the sample data onto the K orthogonal components capturing the most variance. These components are referred to as axes of variation, and their loadings onto the SNPs capture structural allele frequency differences. So you can then visualize the result by plotting the projected data, typically as biplots or scatter plots of 2 dimensions at a time. What do these look like? In the case of discrete structure, groups will tend to appear as clusters. You need kPCs, or dimensions, to fully separate k plus 1 groups, which form the corners of a simplex. So 1D separates 2 groups, 2D for 3 groups, which form a triangle as in this example, 3D for 4 groups, which form a tetrahedron, and you can use multiple biplots for visualization. So you could also plot PC1 versus 3, and 2 versus 3, and 3 versus 4. I encourage you to look beyond the first 2 PCs in practice. In the case of admixture, admixed individuals will fall between the clusters at locations corresponding to their admixture proportions. And fundamentally, this is because allele frequencies interpolate linearly under admixture, and PCA is a linear dimensional reduction. And as for spatial structure, I invite you to look at the well-known and striking main figure from Novembre's 2008 paper entitled Genes Mirror Geography Within Europe. If you want to go deeper on PCA for spatial data, Novembre and Stevens in the same year showed how PCA can even capture general nonlinear variation of allele frequencies with geography. Well, all that's how things would work in an idealized setting with unlimited amounts of data. It's important to remember that PCA is simply a function of the data. In some sense, it's an estimate of some true underlying population structure. But the data are noisy, and you don't have anything like true ancestry-specific allele frequencies, rather just genotypes sampled from them. Estimation and inference from noisy high-dimensional data is non-trivial, and in practice, you might actually not have sufficient signal to resolve all the structure. It turns out there's a critical threshold for FST depending on the data size, actually the number of genotypes n by m, below which the detection of structure becomes impossible. This result is from Patterson et al. For 2 discrete populations, and it depends on the mathematics of random matrix theory. The math actually says a bit more. Even above the detection threshold, the estimated axes of variation may be inaccurate. These issues affect smaller data sets especially, but even in larger data sets, there may be axes of finer structure that remain poorly resolved. You can always run PCA and get a result, and people often look at the eigenvalues of PCA to understand the variance explained. But that's the variance in the sample. It actually doesn't give you a clear lens into the properties of genotype PCA as an estimator of population structure. What's it really estimating? How well is it doing? The answers to those questions shed light on both the way we understand population structure and the way we estimate it in practice. And this is actually research I'm actively working on with Ben and colleagues. I'm just going to pause here to show you this figure from a review article of Hen et al. In 2012 entitled The Great Expansion. It presents a simplified picture of our demographic history as a species at the largest scale. The story is actually quite complicated and uncertain, particularly the more ancient history in Africa. But as we continue to gather genetic data, both present and ancient, we'll continue to infer and refine more detailed histories. Examples include complex admixture events, both ancient, involving archaic humans, the Neanderthals and Denisovans, and somewhat more recent, involving, for example, the ancestral populations of India or Europe. Combined with data, population genetic theory gives us another way of looking into the past. And I'll close by mentioning the relevance of population structure to genetic association studies, or GWAS. So here you seek associations between individual phenotypes and genotypes, as you just learned about in Ben's lecture, and we'll explore in depth throughout the workshop. Now suppose a trait exhibits systematic differences between 2 groups. The average of a quantitative trait like height, or the prevalence of a binary trait like type 2 diabetes. Such differences could occur for both environmental and genetic reasons. If there is population structure, though, then there are also systematic allele frequency differences genome-wide. But that means that any variant that is ancestry informative, or drifted between the two, will appear spuriously associated with the trait. In other words, ancestry Z is a confounding variable, affecting both the genotype X and the phenotype Y, so both the predictor and the response, which leads to genome-wide inflation of association statistics. This phenomenon of confounding due to the correlation of a trait with population structure is known as population stratification. There's actually an elegant solution to this problem, which was proposed by Price et al. In 2006, and it's to include principal components of the genotype matrix, as we just saw, as covariates in the phenotype-genotype regression model. And this approach has proven highly successful and has been almost universally adopted. Unfortunately, as I alluded to earlier, PCA doesn't always fully capture population structure, especially in smaller samples and possibly even with finer structure in larger samples. Those issues may partly explain why some older GWAS results, and even sometimes more recent ones, sometimes still show signs of residual stratification, even after correction using PCs. And even subtle stratification can affect some downstream analyses. Related to the research I mentioned on PCAs and Estimator, we're also investigating the consequences on residual stratification in GWAS. Once you understand how accurate your PCs are, you can estimate the extent of phenotypic stratification along each axis of variation. And finally, how well your sample PCs will control for the resulting confounding in the association analysis. That's all for now. I'm sorry we couldn't be together in Boulder this year, but I encourage you to be in touch with any questions that may have come up for you. And thanks so much for watching.